Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the CAMP and protein kinase A. This is the topic related from the cell biology, and I want to say thank you to all of you for support me a lot, and I also say this is my request to the new ones in my channel. Please do share and subscribe this channel for if this is helpful to you, and obviously after subscription you just hit the bell icon for getting notifications of all my uploading videos. Clear? And obviously you can comment in my comment box for the proper topic, about the topic already I have explained in my channel, so you can obviously set a comment in the comment section. Okay, so let's continue. This is the CAMP and protein kinase A. So here the CAMP, what is saying that CAMP directly activates CAMP dependent protein kinase A. Okay, so this directly activates which one? This activates CAMP dependent protein kinase A. Okay, and the short form of the protein kinase A, this is the pKa. Clear? And this CAMP that induces the opening of certain ion channels in the plasma membrane of some highly specialized cell. That means in case of, not for all cells, only in some highly specialized cells, this CAMP help to open the ion channels. Okay, and pKa, that is the protein kinase A, this catalyzes the transfer of the phosphate group from ATP to the specific target proteins like serines or threonines. Clear? So CAMP, what is the CAMP role? So CAMP's role is that it induces to open the ion channels in the highly specialized cells. And what is the role of the pKa? This catalyzes the transfer of phosphate group from ATP to specific target proteins. Obviously, example is that serines or threonines. And in that way, they can regulating their activity. Whose activity? The target protein's activity is regulated by this pKa. Okay, and obviously in that situation CAMP has an important role. So in the inactive state, pKa, suppose this is pKa, this consists of a complex of two catalytic subunits and two regulatory subunits. Okay, suppose this is pKa, this is a brief sketch, and this is a complex, and it consisting of two regulatory subunits and two catalytic subunits. Okay, and the binding of cyclic AMP or CAMP to regulatory subunits. So binding of CAMP, this is CAMP, to the regulatory subunits. What happening in that situation? This alters the conformation, causing them to dissociate from the complex. Okay, so when CAMP that is to be bound with the regulatory subunits, then what happening? The conformational changes occur. Okay, conformational changes occur, and that's why the catalytic subunits that cannot remain continuously bind with the regulatory subunits. So here the dissociation of the catalytic subunits and the regulatory subunits is seen. And here this, after just separation of from the regulatory subunits, this catalytic subunits tendency is to enter into the nucleus from the cytosol. Clear? And this released catalytic subunits thereby activated to phosphorylate specific substrate protein molecules. So now this, what is the function of this when it enters into this nucleus? its function is to activate the phosphorylation process in some protein molecules. Here the protein molecules is CREB. Before entering the catalytic subunits, it remains in active state, but when it enters, it transforms into active state. Okay, this transform into active state, and here the phosphorylation of the protein that is done by the catalytic subunit, or you can say that catalytic subunit induces the phosphorylation of these protein molecules. And active catalytic subunits, it can phosphorylate a number of target proteins. They may be cytosolic or may be nuclear, and they remain obviously or must be attached with the serine or threonine residues. Okay, in that proteins they try to active on that proteins or on that target proteins who are already attached on the serine or threonine residues. Okay, and here CREB, it is the full form. Okay, it's raining in the outside. Okay, so some sounds may be enter in my video, so you just adjust with that. And CREB is that what is that CREB? CREB is that CAMP response element binding protein. The full form of the CREB is that CAMP response element binding protein, and which is activated by the phosphorylation. Okay, without phosphorylation they cannot be activated, and if they activated by the phosphorylation, what is happening? Stimulate the gene transcription. This stimulate the gene transcription. Okay, and here is another thing is say that phosphorylation at a single serine residue greatly increases the activity of the CREB. Okay, so if phosphorylation of single serine residue is occurred, in that situation this increases the activity of the CREB bound to CRE. CRE is what? CAMP response element. Okay, so one single serine, phosphorylation of one single serine, that greatly increase the activity of the CREB bound to CRE, and which is found in genes and whose transcription is induced by CAMP. So here the transcription of the gene that is induced by CAMP. So this is very short and brief note where the role of CAMP and PKA, these protein kinase A, these are described here. Okay, because without CAMP the transcription is not possible. This is induced by CAMP. Normally this is occurring, but this is induced. CAMP, the CRE's full form is CAMP response element. That means CAMP is involved here, and if CAMP is present there, this process is induced by CAMP. Okay, so I think you can properly understood the topic, that is CAMP and the protein kinase A. So in my next video I will try to explain about another short topic related to obviously cell signaling. Okay, so continue with my channel. Thank you.